Okay, so today, functional network of novel Barley microRNAs and their targets in response to drought. As I promised you to show you our results connected to drought stress, microRNA actions, and then heat stress will be later. So today, drought. So I will first describe, as I told you yesterday, that microRNAs regulate plant development and adapt to plant stresses. So if you see here, you may have different stresses like drought, heat, darkness, UV light, oxidative stress, pathogen attack. And all these stresses induce microRNAs down or upregulation, affecting physiological adaptation and molecular adaptation, as well as senescence of plants. That's what we talked yesterday. So I will describe you now an experiment we did. So we grown plants first in greenhouse, but then we decided to go to the growth chamber. Why? Because in the greenhouse, you can count, of course, the number of plants per pots, but you have limited control of light and temperature. Also, soil composition and watering are controlled. However, and if you see, the drought affects plant growth. However, when we went to the growth chambers, you may see that light, day lengths, and temperature are fully controlled. So we just see the drought response, and then you can see control and drought plants that are, that drought also indeed affects plants when they grow in growth chamber. So what was our experiment? We took plants that grow in the conditions of 70% of soil water content. Is this soil water content known for you, this parameter? Or should I explain it? Okay, so it is like first you took jak to powiedzieć, ziemię, jak to powiedzieć. You took the soil, then you heat it to do it completely dry, and you wait it, and then you add water as long as it goes through. And then you know what is the content of water, and this is the 70% of soil water content, yes? And then you can wait after a couple of hours, after 1 day, 2 days, your pot, and then you will know how much water disappeared from your pot. So this is the conditions that first we had 30% of soil water content. It was a 1 point. This is shown here. And this is where generally already the plant starts to sense the lack of water. Then we have 20% of soil water content. This is shown in yellow because this is extremely low water content for plants. And then even 10% of soil water content. And when it reached 10% of soil water content in the pot, we re-watered plants. And then we checked points like 6 hours after rewatering and 24 hours after rewatering. For NGS analysis, looking for conserved microRNAs first of all, and I will tell you about conserved because looking for conserved, we discovered a new one. So another one microRNA. So we decided to take 2 points. This is this 20% of soil water content and 6 hours after rewatering to do small RNA sequencing using Illumina and to look for conserved microRNAs. So what we found is that this is a summary of changes of conserved microRNAs accumulation in drought or rehydration treated plants. So generally 90% of conserved 90 microRNAs were not affected by drought. 45 were downregulated while 15 were upregulated. Now we looked what happened with these microRNAs of these 3 categories after rewatering. And after rewatering, of these that were upregulated, 9 remained upregulated, 6 were again as in control, so they went down. And out of 90 not changed, 7 were downregulated, 3 were upregulated after rewatering. So I have to stop. Okay. Okay. Yes? Okay. So, and then, should I repeat this slide or it's not necessary? Okay. So then when we regarded these 45 that were downregulated, 30 of them came to normal level, while 15 still remained downregulated. So we generally concluded that the observed changes of microRNAs accumulation induced by drought are substantially reversed within 6 hours of rewatering. So it's enough. So then we looked for microRNAs and their targets. MicroRNAs, I will just generally show you, don't worry, for microRNAs and their targets. And before I will come to that, I think I should explain you how we identify targets. And we identify it using so-called parallel analysis of RNA ends. It's PARE, P-A-R-E, parallel analysis of RNA ends. And what do? First of all, you isolate your poly A plus RNA. You discard all kept connected RNAs. So full RNAs you discard. You are interested only in degradation products. When you have your degradation products, you do cDNA, and then you cleave it with a specific nuclease that binds to cDNA, but cleaves, let's say, 40, 50 nucleotides upstream. And then you add adapter, and you look for 5' ends of your degradation products. Is it clear? Okay. So now you have, we say, degradome library. So if you know where your microRNA cleaves, you would expect the enrichment of one 5' end of your cleaved RNA, let's say now cDNA. Yeah? So if you have a high peak of a given 5' end of cDNA, but the earlier mRNA, and it is just in a position where you know from bioinformatic analysis microRNA should cleave, then you may say this is a potential target of microRNA. Of course, we say always potential because you have to be cautious. Not always you get, very clear pictures of one sharp peak and then only small peaks. Sometimes it is within a quite number of peaks. So then we need to confirm it with other approaches like 5' rays you are aware of, probably, yeah? No? Okay, 5' rays is a technique where you take your poly A plus RNA, you reverse transcribe it to cDNA, and then look where your 5' end is in your cDNA library, yeah? And if, let's say, out of 20 clones, yeah, you isolate your band, what you got, and you estimate how big should it be. You can elute it. Sometimes people do not elute that, go directly to cloning. And then you sequence clones and look how many clones you sequenced, how many had 5' end exactly where you expect your cleavage site should be. Is it clear? Mm-hmm. So then 5' rays is another technique to prove that you have indeed a cleavage site for microRNA. So if you prove it nicely with two techniques, it's fine. And on top of that, you can look whether, if you downregulate your microRNA, whether the target is going up, or when your microRNA is upregulated, whether your target goes down. So everything together gives you somehow an idea whether this is a real target for microRNA. Okay, so these here I show as an example. This is we call it T-plot. So what you see it here. Here you see the length of your mRNA. So from nucleotide number 1 till the very end. And here you see the number of reads per 5' end. And you can see here you have a lot of different enrichments in 5' ends, but one is high and clear, and is exactly where you should expect cleavage site. So this is quite nice. This one is not so nice already, but still you can see this is a conserved microRNA's known target, but how it can look like among different degradation products. This is the cleavage site when we expect it exactly to be. So we call them T-plots. So what you can see here now, so you see here we have microRNA counts from NGS in drought after rehydration and their targets identified, how they look like. So in the first group you have the microRNAs, conserved microRNAs we identified are upregulated in drought, upregulated after rehydration. And then you see that targets identified are downregulated in drought and are downregulated also after rehydration. And when you see a red dot, it means this is a novel target for barley at least. Maybe it was announced already for other plants, but never announced for barley. And among them, we have also novel, completely novel targets as well. So the second group is where microRNA is upregulated in drought, but is down, let's say, goes to the normal level after rehydration. So you see that selected targets go down in drought, but are not changed after rehydration. And these are, with black dots I marked, these targets I will talk later about, and these are conserved for 172, apetala-2, transcription factors, repressors of flowering we were talking yesterday about. So the next group is about microRNAs that do not change in drought, but are upregulated during rehydration. So the targets are not changed in drought, but downregulated in rehydration. One of them is novel for barley. Then we have a group of microRNAs that are downregulated in drought, downregulated after rehydration. Their targets are upregulated in drought and after rehydration. And here we saw also quite a lot of novel targets for barley. And then the last group are microRNAs that were downregulated in drought, are not changed after rehydration, so they go back to the control level. And then these targets are upregulated. And here we see also some novel targets, and this is a quite novel target I will talk about in a moment. So what you can see, and by the end we have a small group of microRNAs that are not changed in drought, downregulated after rehydration. You see the same, that it is not affected in drought, but is upregulated in drought, after rehydration, and one of these targets is novel for barley. So we focused on one selected example, because it was really interesting for us. And this is a gene that encodes 172 microRNA. I should also mention there are many members of this family encoded within the genome of barley. And only this one, only microRNAs derived from this one, and we are lucky that we are able to discriminate them by sequence, are up or down regulated. So what we found is that from the 5' end, you get microRNA 172b, and while from the 3' end, we see microRNA that we called 5' 172b, and this one we called 3' 172b, because it is from the 3' end, and this is from the 5' end. So what you can see now for this 5' end, so this one, so it is, when you see NGS data, this is downregulated in drought, goes a little bit up after rewatering, confirmed by Northern. So you see that here you can, it's faint, but you see microRNA here. It's not visible in drought. It is in control after rewatering, and it starts to appear after rewatering as well. And this is this microRNA 172p, 3p. And you can see that it is upregulated in drought. It is also upregulated, but not significant, is statistically not significant after rewatering. And you can see it is upregulated here in drought. So you have one precursor, the reads are almost the same, but the level of the precursor is not changed, but the level of microRNAs changed. Yeah? So in drought, the one is going down, but the second is going up and is reversed in controlled conditions. So this is something unusual, because normally we know that sometimes people see passenger microRNA stars appearing in some stresses as a microRNA, still sometimes not known function, but that two microRNAs may originate differently, are regulated in their post-transcriptional processing in response to some environmental cues. This is something really original. And for 172.3p, it's a very well-known microRNA that targets apetala-2 like mRNAs to cleavage. So here you can see these beautiful, beautiful T-plots where this is clear, only one band, and around almost nothing. And this is this microRNA, 3p, that targets these apetala-2 mRNAs. The second end, we know that apetala are repressors of transcription. And it is also, I think, understandable when plant encounters drought, it starts to speed up flowering processes to give seeds and to stop its vegetation. So what you can see, once again, the microRNA is upregulated and in drought, and all targets are downregulated. So how can we see that barley was flowering earlier? It is very difficult because barley exhibits so-called kleistogamy. Do you know what is it? Oh, you know, okay. Yes? Everybody knows? Okay, so everything goes inside. So what you can only measure is this caryopsis, so young grain, and its width. So when fertilization and flowers were earlier, the width would be bigger. When it was late, the width would be smaller. So what you can see here, these are caryopsis, these are anthers, isolated. So here is wild-type plants. So width is quite narrow, while in drought, they are much brighter, what means that plants indeed started to flower earlier in drought conditions. So what about the second one? For the second one, we found also a target, but this is not so nice as you would expect, because it is at the very 3' end of mRNA. It's also unusual because generally, as I said yesterday, microRNAs in plants mainly attack coding sequences. But here it is almost at the very, very 3' end. So we had to prove it. But since it is at the very 3' end, it was quite difficult to get these small fragments from here. But we managed to get 4 clones out of these 3 finished exactly at the place where we should expect microRNA should cleave, and 1 was downstream. And we do not care a lot about downstreams because, you know, mRNA after cleavage is rapidly degraded. So it is more suspicious when you get 5' raised fragments that are upstream of your cleavage site than downstream of your cleavage site. So confirming this, we proceeded further with our experiments. And what we did, as you may see here, microRNA, this 5' is down, 5' is downregulated. mRNA for trehalose 6-phosphate synthase is upregulated. So there are available antibodies against this enzyme, trehalose 6-phosphate. So we found that in drought, it is more than 18-fold upregulated. So it is in agreement with what we see. And after rehydration, it goes down, what is also in agreement. Then we measured also trehalose level, and you can see that during drought, trehalose level is strongly upregulated, while it goes down after 6 hours of rehydration. So this is extremely speed process. And now how the trehalose is synthesized. you get UDP glucose, you have glucose 6-phosphate, and then the enzyme trehalose 6-phosphate synthase produces trehalose 6-phosphate. This is also an important molecule, signaling molecule for plants, rather in small amounts because it's rapidly turned by trehalose 6-phosphate phosphatase to trehalose. And we did not measure trehalose 6-phosphate because it is toxic for humans. So we stopped, we didn't do that. We just measured trehalose. But we know also that if you have more trehalose, probably you have also more trehalose 6-phosphate. And we know that these molecules are really important for plants. So if you see now, trehalose is shown in orange boxes. Trehalose 6-phosphate is shown in green boxes. And blue boxes show both action of trehalose 6-phosphate and trehalose. So drought and trehalose is stabilizing effect on proteins and membranes. It also affects insect and pathogen defense, and it also affects rhizobial and mycorrhizal symbiosis, while trehalose 6-phosphate is involved in aging, is involved in embryogenesis, in photosynthesis efficiency, also in photoperiod pathway, and also a starch to sucrose conversion, so that's a lot. And both of them regulate also stomatal aperture, both of them. So what you see here is just to show you that the mutant of trehalose 6-phosphate, the gene that whose mRNA is cleaved by 172 5P, has a late flowering phenotype, what means that this trehalose 6-phosphate is also required to regulate flowering of plants. And here just I show you how trehalose is produced. First you have trehalose 6-phosphate, then from 2 glucose molecules, then you have trehalose 6-phosphate that can be then also turned to glucose. So we know that desiccation tolerance results from massive accumulation of trehalose or sucrose. So it was not known what is accumulated in barley, but now we think in barley trehalose is the main sugar that is accumulating. And both trehalose and sucrose are non-reducing disaccharides, chemically less reactive than most other naturally occurring sugars, can be accumulated to high concentrations within cells, damaging and within cells without damaging cellular structures, and they stabilize effect on proteins and membranes in vitro, particularly trehalose. While trehalose additionally protects cells by initiating controlled autophagy, thereby avoiding uncontrolled damage that would otherwise trigger programmed cell death. So trehalose, we think, is now also controlled, tightly controlled by microRNAs from this one gene that also produces 172.3p, which targets apetala-2. So this is an example of a plant that accumulates up to 95%, it survives up to 95% of dehydration by accumulation of sucrose. And this is another plant that accumulates trehalose during dehydration. So summarizing these results, we can say that response to drought via microRNA 72b 5p3 results in earlier flowering and osmoprotection. So we have drought, microRNA 172 BP is downregulated, trehalose 6-phosphate synthase is upregulated. You get more probably trehalose 6-phosphate. You get more trehalose and osmoprotection to drought stress. While from the other stem, microRNA 172.3p is upregulated and downregulates apetala-2-like proteins and speed up flowering of plants. So this 5' 5' P172B is a novel microRNA that is quite active during drought. So now, what about other microRNAs in barley? This is not our work, that regulate drought stress tolerance through tomatal density. For instance, microRNA 393 negatively regulates drought stress tolerance through stomatal density. Here you have 3 mutants of barley, wild type, overexpressing 393, and then this is target mimics. It means you introduce a target that has a complementary sequence, but it cannot be cleaved of microRNA. Then it acts as a sponge, attracting microRNA that cannot act, right? Yeah. So what you can see in mutants, that this is abaxial site of the blade. So you have here wild type stomata. Here you have overexpressing mutants that they are shorter, and here in downregulation you have back longer stomata. And when you compare the widths, the length, of stomata, you can see the wild type, then downregulation in the overexpressing mutants, and this is the mimic mutant. So what now you can see, these are all 3 plants, wild type, overexpressor, and mimic. In control conditions, they do not change a lot between each other, while in drought, the overexpressor is dying. So it does not withstand drought. This one is wild type is withstanding, however, to much less extent than the mutant where 393 is downregulated. So what about 393? So it is induced by abscisic acid, so let's stress hormone, yeah? If you have a lot of 393, then you downregulate auxin signaling. As an output of this, you get the less number and less of stomata, and more stomata, but they are smaller, and you get drought sensitivity. And you get drought sensitivity. Is it clear? Okay. So before I will go to the next part, talking about novel microRNAs, I just want you to advertise a web page we constructed for barley conserved MicroRNAs expression profiles that can be found in the MIREX2 database. And this is the web page. And you have here not only MIREX, but barley, but you have also data for Arabidopsis and for a liverwort, because we are also working on the earliest land plants. This is also for liverwort from genus Pellia. So this is a web page you can easily access and look for some interesting data, I think. This is the update. The first issue was published in Nucleic Acids Research, this in BMC Plant Biology. So we have 3 plants. This is Arabidopsis, barley, and this liverwort. And you see here that we have, I don't see it, sorry, that we have here, I don't see it either. These are probably microRNAs, the number that we have, developmental stages, mutants of microRNA biogenesis, and this is, these are NGS samples, so downloaded NGS samples. This is what you have at the web page. So if you enter the web page, for instance, for barley, you can select developmental stage of barley. And then if you select developmental stage, you can go to microRNAs, you can go to the precursors expression, and eventually to mutants. In the case of barley, there are no mutants. But also, if you click here, this developmental stage, you can go to degradome data. And these are degradome data for barley. So if somebody is working with grasses, this could be advantageous to see whether microRNA is acting also on some mRNAs that are maybe evolutionary conserved between grasses. So now, in search of novel microRNAs responsive to drought. So the data I was talking about looking for conserved microRNAs were also used to look for novel microRNAs. So how do we do that? This is just a general view of NGS data where you just show the frequency of a given length of small RNA that is represented in your library. And here you can see two large peaks. This is 21 and 24. This is classical for microRNAs, and 21 and 24. And this is how NGS data should look like. And then what you do, you have your sRNA reads. You then take cDNA library or reference genome. Reference genome is better. Then if you have a reference genome, it's much better. Then you discover clusters, mapping sRNA reads to Barley, cDNA, or genome. Then you quantify and do phasing analysis. Phasing analysis means that you will expect that one species is overrepresented over the others, while phasing will you do small RNAs along the gene or along your cDNA. So phasing is not typical for microRNAs, so you look for some that have this peak. Then you look for, you retrieve sequences flanking the clusters. So because you look for microRNA, you want to see at least stem and loop structure. And then you perform RNA folding and hairpin identification. If your sequence is able to fold into a typical hairpin for microRNAs, and then identifying a microRNA in the cluster and microRNA star, and then we usually do Northern blot to confirm that this is indeed a single molecule. We are old-fashioned and like always to see hybridizing molecules. This is much more convincing for us. So these are novel microRNAs we discovered. So this we called Hordeum vulgare X9A, and this is the same family, X9B, so two microRNAs differing in one nucleotide. But you can see nice reads and nothing else, almost nothing else. So the cluster is really pure, only 21 nucleotide long protein. But if you can read here you have 2,500,000 reads, while here you have 12,000 reads. 12,000? Yes, 12,000 reads. So the B is much less expressed than A. So we did two Northerns, but do not believe in this one. In this one, we see this one. Yeah. So if they differ by one nucleotide, you cannot discriminate them in Northern blot. Then you need deep sequencing, yeah, to see single nucleotide resolution. So, and you see, these are nice peaks of 21 nucleotide long molecules with a hybridization signal. Then X11, that is also found at 21 nucleotide lengths and is present during hybridization. 13, which is also nicely present in hybridization and at the 21 lengths. Then here you can see a little bit more of these cluster companions, but 21 is dominant, and here is 21 nucleotide long molecule, and X10 again is also present here, and 21 long nucleotide molecules are present here. So then we looked for stem loop structures for all of them. So this is for 9A, this is for 9B. So although they represent one family, they differ in their structure of precursors, because here you can see it is much longer. Yeah, we were not able to fit it into the slide, so it is much smaller. Can you see it? Yeah, okay. And this is the gene structure. So the stem and loop structure is found within second exon of microRNA gene. This is microRNA. This is microRNA star passenger. This is the second one Here, the reverse, the gene is found, microRNA stem and loop structure is found in the first exon. Here, the gene is intronless. Here, the gene contains 2 exons and stem and loop structures in the second exon. And here's again a very interesting situation that we have 2 microRNAs detectable by northern and by deep sequencing data, not differing much in counts, and derived from both arms. So maybe this is something that we should look in Poaceae family, in grasses. It's not so common, at least for Arabidopsis. For other plants, I forgot now the name. So I think, who knows? Who knows? Maybe this is something really happening in grasses. So we looked for, and maybe I should mention that all these hairpins are classical hairpins for microRNAs. So we were confident that we identified novel microRNAs. So we looked for targets of these microRNAs, and we found that this is a target. This is disease-resistant protein, and very nice diploid. This is mitochondrial transcription termination factor, mTERF. Here also quite nice diploid. Here is protein belonging to serine protease class. Not very nice, but still taken into account because of further experiments I will show you. And this is protein with CS domain involved in protein-protein interactions. Here, one nice signal. And here is protein with zinc finger and SWIRM domain. This is in a domain suggests it is involved in chromatin remodeling. So then we looked for the correlation between microRNA level and identified target. And what we found, this microRNA 9A and 9B, they of course target the same mRNA, but you can see here that in severe drought, like 20% of soil water content, microRNA is dropping strongly down. MicroRNA is dropping strongly down, while its target, sorry, it should be here. This is my mistake. This is control. This is upregulation of target sequence. So again, when you see microRNA in northern and drought, you can see it is downregulated. And what about this disease-resistant protein? We have no idea. We will write now projects concerning particular cases to look in transgenic plants when we overexpress microRNA, downregulate microRNA, overexpress target, downregulate targets, to see what will happen in drought, yeah? So this is still open question because I am showing you now results that will be continued. So we have drought, microRNAs is downregulated, disease-resistant protein is upregulated. What about drought? We have no idea, but probably the fungal and viral pathogens somehow maybe are also more sensitive because plants affected with stresses turn to be weaker. So I would say they may be also more susceptible for some pathogens and insects. So the other one, X10, is again downregulated. Its target is upregulated. X8 is downregulated. mRNA is upregulated. I do not show you here northerns because in drought they were not detectable. They were not detectable. We were able to detect them in control conditions, but not in drought. So again here, this target is protein with CS domain. So this is somehow involved in the recruitment of heat shock proteins to multi-protein complexes. Maybe this could be a kind of adaptation to drought. Who knows? Looks interesting. And this one is drought. We have microRNA X8. This is zinc finger protein with the SWIRM domain. So maybe changes in chromatin organization, sensitivity to abscisic acid, and changes in gene expression in response to drought has to be checked. And the next one is X11. it is downregulated. Here we see the downregulation. The target is upregulated. The targets are MTERFs, mitochondrial transcription termination factors, we are actually interested in. They are called mitochondrial transcription termination factors, but they are also present in chloroplasts. And these are from chloroplasts, so the name is misleading. But their action in drought, this is completely unclear. So we will see what will happen when we will up or down regulate its expression in barley. And then microRNA 13, that is downregulated in drought. Look, majority of these microRNAs conserved were also downregulated. Do you remember from the beginning? And these all are also downregulated. And the target is upregulated. So this is serine protease class proteins. Maybe this is a more higher proteolytic activities, somehow affecting drought sensitivity. We do not know it. So I missed here one slide, sorry. This is what I wanted to tell you. So during our studies, somebody already discovered X13 and published a paper about it. So this 13 is done. So we know that in control conditions where you have wild type or a knockout of microRNA or overexpression, in drought, wild type, but particularly knockout, is dying, while microRNA overexpressing plants are withstanding drought. And this was done in Golden Promise variety. This is under other promoter ubiquitin here, 35S, and the same result. And here you can see that this is another variety, but the result is the same. So upregulation of this microRNA may render plant to be more resistant to drought. So we are eager, what about the others? Sorry, I don't know what happens. So now this is even less advanced, what I will show you now. So these are further novel microRNAs derived from different barley growth stages and mutants, but not from drought experiment, but we have some other barley mutants we are interested in. This is called Whirly. Whirly1 is a protein that is present in chloroplast and in the nucleus, evidently playing two different roles. In nucleus, acting like rather transcription factor, we are working on it. And we also analyzed microRNA pool from mutant and from wild-type plants, and were able to discover novel microRNAs, so we immediately checked whether some of them maybe are drought responsive. So this is how the analysis looks like. So this is example of a cluster generated with the usage of NGS and small RNA data and barley genome. What you can see here is a cluster showing this. I think you are aware this is complementarity. Here there is no complementarity. Here is microRNA that has 358 reads, while this one, I don't see it, has much less. So this is star, this is main microRNA, and these are these clusters of microRNA around and microRNA star, but with less reads. So this is like it looks like. So then you can fold a stem and loop structure, and this is here. And this is calmodulin-like, and this is EF hand calcium binding protein. Where is here here is the signal. So it has something to do with calcium signaling and regulation. How about drought? We put question mark, but in our drought NGS data, we see it, but it is not up and not down regulated. So it's not affected. So this one, we identified. We also identified a very nice T-plot. This is pre-mRNA cleavage complex 2. So this is poly-A tail addition, so we can have alternative polyadenylation sites. So mRNAs can be shorter, can be longer within in frames of their 3' UTR. Nevertheless, in drought, we do not see change of this microRNA. So we will look at it in the aspect of Whirly mutant, but not drought. This is what I told you already. This is now another microRNA that has a target, and this kind of target must be conferred by 5' race. And we still didn't do that. And, but microRNA is there, but it's not upregulated, not downregulated in drought. And this one, this is another cluster, another nice microRNA. This is a eukaryotic aspartyl protease family protein. So it has expression of genes related to ABA signaling and synthesis, and stomata index and stomata density. Could be interesting, but is not affected. Its amount is not affected in drought. But the last one, that we have no target for it, so we have to look for it more carefully now, is almost 40 times downregulated in drought. But I can say anything more because we know only this is just last result I got from my postdoc, that it is downregulated, so we will for sure look for this microRNA as well. And this is everything I wanted to show you, maybe some conclusions, that one microRNA precursor may contain two active microRNAs with opposite accumulation pattern during stress, that trehalose seems to be an essential sugar in drought tolerance in barley, that many identified non-conserved microRNAs are responsive to drought, targeting mRNAs yet often unknown as important in drought response, and that there is still a space for search for novel microRNAs, target modules, with important functions in crop plants in drought resistance. And with that, I would like to thank you. And this is just what I didn't do. I have it on my computer. I was showing you